Hello everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to share with you some insights on a fascinating topic. The focus of our discussion will be on the source of contamination, specifically the human microbiome. Over the course of this presentation, we'll explore how our own microbial communities can influence contamination, and what that means for health and hygiene. So, let's dive in and discover what lies within us. Let's start by understanding what the human microbiome really is. It's essentially a collection of microorganisms that live on and inside our bodies. For example, our skin has its own set of microbes, like Staphylococcus species, which are quite common. We also have microbes in our respiratory system and in our gut, which play important roles in our health. These microbes are not just static. They're constantly shed into our environment, meaning they can be transferred around and potentially contaminate surfaces or equipment. Now, when we talk about clean rooms, which are designed to control air, surfaces, and equipment, it's crucial to remember that humans are actually the biggest source of contamination. Despite all the measures we take to keep these environments sterile, personnel can contribute up to 80 to 90% of microbial contamination. So, understanding the human microbiome and its behavior is fundamental in maintaining the integrity of these controlled environments. It highlights why human hygiene and behavior are so critical in our overall contamination control strategy. Now, let's take a closer look at the regulatory perspective outlined in Annex 1. One of the key points here is that personnel are considered the primary source of contamination risk in manufacturing environments. This means that controlling the presence and activity of staff is crucial to maintaining product quality and safety. To address this, regulations require that we minimize personnel presence as much as possible, especially in critical areas. When staff do need to be there, strict gowning procedures and hygiene practices are mandatory. This helps prevent any contamination from skin, hair, or clothing from entering the cleanroom environment. Additionally, access to clean rooms must be carefully controlled. Only authorized personnel should enter, and often, this involves controlled entry points and protocols. All these measures are mandated by Annex 1 to ensure that contamination risks related to personnel are kept in check. Ultimately, these controls are not just about compliance. They are about safeguarding product integrity and patient safety, which is at the very heart of the overall manufacturing philosophy. Let's take a closer look at how microbial contamination occurs in cleanrooms. The human microbiome is a primary source of contamination, and it can affect even the most carefully maintained environments. One of the main ways this happens is through shedding of skin cells. As we move around, tiny skin cells are constantly released into the environment. These skin cells often carry microbes attached to them, which can then become part of the particulate matter floating in the air. Microorganisms don't usually travel alone. Instead, they hitch a ride on these particles. When we move through the cleanroom, these contaminated particles can become airborne and disperse throughout the space. This airborne dispersion is a key factor in how microbes spread within the environment. Once airborne, these particles can eventually settle onto critical surfaces, such as equipment or sterile areas, leading to potential contamination. It's important to understand that microbes rarely travel independently. They're almost always attached to particles, which makes controlling their spread more challenging. So, in essence, human activity and movement are central to microbial contamination, with particles acting as vehicles for microbes to reach and settle on sensitive surfaces. Recognizing this mechanism helps us develop better strategies to minimize contamination risks in cleanroom environments. Let's talk about respiratory contamination and its sources. When people talk, breathe, or cough, they can release particles into the air. These particles, known as aerosols, can carry microorganisms such as bacteria and fungi. This means that everyday actions like talking near sensitive areas, for example, a filling line, can introduce microbial contamination. It's a natural part of how humans interact with their environment and can impact cleanliness and safety standards. So, understanding these sources helps us better control contamination in our processes. Let's talk about the role of human behavior in contamination. When we move around, it actually increases the amount of particles we shed. For example, walking can result in about 10 times more particles being released into the environment. This shows how movement itself can elevate contamination levels. Poor aseptic practices also play a big role. Touching sterile surfaces or making fast, careless movements can transfer microbes and compromise cleanliness. So, our behavior really has a significant impact on contamination levels. Now, let's look at some specific sources of microbes from the human body. Hair can carry microbes, acting as a sort of microbial reservoir. Sweat provides nutrients that microbes love to feed on, which can encourage their growth. Sebaceous secretions, or sebum, contain oils that support microbial proliferation as well. Microbes are shed from various parts of the body, mainly the skin, eyes, and mucous membranes. These are key sources for the human microbiome, which is the collection of all the microbes living on and inside us. Understanding these sources is crucial because it highlights how human presence and behavior can influence microbial contamination in our environment. Now, let's consider the impact on sterile products when contamination occurs. Contamination can lead to several serious consequences, starting with product sterility failure. When sterility is compromised, the product may no longer be safe for use, which can have dire effects. This often results in batch rejection, meaning the entire batch has to be discarded, leading to significant financial losses and delays. Most importantly, contaminated sterile drugs can pose a direct threat to patient safety, increasing the risk of infections such as sepsis, which can be life-threatening. Moving on, microbial contamination in sterile drugs is not just a manufacturing issue. It can be life-threatening for patients. These microbes can come from various sources, and one major pathway is airborne transfer. Microorganisms become airborne through processes like particle resuspension, when dust or particles containing microbes are disturbed, and airflow disturbance, which can spread microbes throughout the environment. It's also crucial to understand that personnel play a significant role in airborne contamination. In fact, they are responsible for up to 80% of airborne microbial contamination in controlled environments. This highlights the importance of proper gowning, hygiene, and aseptic techniques to minimize the risk of contaminating sterile products and ultimately protect patient health. Now, let's take a look at monitoring and detection, which are crucial components in maintaining a safe and compliant environment. First, under environmental monitoring, or EM, we have several key methods. Air sampling is used to detect airborne contaminants, helping us understand what particles or microbes might be present in the air we breathe. Settle plates are simple but effective tools. They allow particles to settle naturally, giving us insight into surface contamination levels. Surface swabs are another important method, as they help us identify microbial presence directly on surfaces that are frequently touched or critical to our processes. Moving on to personnel monitoring, we focus on how we can track potential contamination from staff. Glove prints and finger dabs are common techniques here. Glove prints can reveal whether contamination is transferring from personnel to surfaces, while finger dabs can help detect microbes on individual fingers, providing a more detailed picture of personnel-related risks. It's also important to note that Annex 1 emphasizes a risk-based approach to personnel monitoring. This means we tailor our monitoring strategies based on specific risks, rather than applying a one-size-fits-all method. By doing so, we ensure our monitoring efforts are both effective and efficient, aligning with overall good manufacturing practices and regulatory expectations. In summary, these monitoring and detection methods form the backbone of our contamination control strategy, helping us identify and mitigate risks proactively. Now, let's take a closer look at the control measures we implement under the regulatory CCS approach. First, we have sterile gowning systems, which are essential for maintaining a clean environment by preventing contamination from personnel. Along with that, restricted personnel access ensures that only those who are properly trained and authorized can enter the clean areas, minimizing unnecessary exposure. Aseptic training is another critical element, making sure everyone involved understands the strict protocols to keep processes sterile. Hygiene control is also vital. Rigorous hygiene practices help prevent any potential contamination sources. Overall, we limit access to only the minimum personnel necessary in these clean areas to reduce risk further. Moving on to our advanced control strategies, we use isolators and RABS, which stand for Restricted Access Barrier Systems. These technologies are designed to significantly reduce human intervention, thereby lowering the chance of contamination. Automation of aseptic processes is another key strategy. Automating critical steps helps ensure consistency and sterility. We also incorporate real-time bioaerosol monitoring, which allows us to detect any microbial presence immediately, so we can act swiftly if needed. For example, isolator systems are particularly effective. They create a sealed environment that drastically reduces human contamination risk, providing an extra layer of protection. All these measures and strategies work together to maintain the highest standards of sterility and safety in our processes, aligning perfectly with our overall goal of ensuring product quality and patient safety. Let's look at this case study to see how contamination issues can arise and be addressed. The scenario here involves aseptic filling contamination, which is a critical concern because it directly affects product safety and quality. The core problem was microbial growth in the finished product, which is a serious issue because it can lead to safety risks for patients and product recalls. Upon investigation, we found that the root cause was improper glove disinfection. This highlights an important point. Even small lapses in hygiene practices can have significant consequences. The impact of this contamination was batch rejection, meaning the entire batch had to be discarded, resulting in financial loss and potential delays in production. To correct this, the team took several actions. First, retraining staff on proper hygiene and disinfection procedures was essential to prevent recurrence. Then, increasing personnel monitoring helped ensure compliance with these protocols. Lastly, updating the CCS, or Control and Compliance System, provided a more robust framework for ongoing oversight. This case underscores the importance of vigilance at every step of the aseptic process. It also reminds us that continuous training and system updates are key to maintaining product integrity and safeguarding patient health. Let's wrap up with some key takeaways. The human microbiome is actually the largest and most unpredictable source of contamination in cleanrooms. Because we can't completely eliminate it, our focus must be on controlling it effectively. That's where behavior and training come into play, making sure everyone understands their role and follows proper procedures. Gowning and monitoring are also critical, helping to minimize the microbes we bring into the environment. Additionally, incorporating CCS-based risk management allows us to proactively identify and mitigate risks. Remember, the success of a cleanroom isn't just about advanced technology. It really depends on how well we control human factors. Ultimately, managing human presence and behavior is what keeps the environment clean and ensures quality. For further support, please don't hesitate to reach out. We're here to help you implement what you've learned today and to answer any questions you might have. Remember, the goal is to ensure you feel confident moving forward. If you need additional resources or guidance, contact information is provided. Thank you so much for your time and attention today. We truly appreciate your engagement and commitment to this topic. We look forward to supporting you in your journey.